We're honored to have with us a man who is actually heading the reproductive biology laboratory at the Camel Reproduction Center in Dubai. He's credited with creating Injaz, the first camel cloned in the world. Please give a warm welcome to Dr. Nisar.
Thank you very much. Okay. So before I go to the actual methods we adapted for creation of the creation of Injaz, the world's first cloned camel, I wanted to quickly, within a couple of minutes, go through the terminology I'll be using quite frequently within the next 30 to 40 minutes in this presentation. So as I should be expecting all of you knowing about what is cloning, and cloning you all might be knowing is the creation of the organism that is an exact genetic copy of the other one, whether it's an animal like a cattle which have been cloned, it's the camels which we have cloned recently here, or whether it is a plant. So the question arises then, does the cloning occur naturally? The answer is yes and no. Yes, for some microorganisms, which, like for example, we have the unicellular microorganisms like amoeba, where the nucleus of this amoeba, it makes a replicate copy of itself, divides into two, and then the cytoplasm of this amoeba divides, and what we get is two daughter cells which are exactly clones of each other. This cloning also we use in our horticulture by in the plants, where we use the cuttings of these plants for their propagation, and what we get is the new generation of the plants are the clones of the original plant from where we have taken these cuttings. But this cloning does not occur naturally in case of the higher animals and mammals. So for that purpose, if we need to clone an animal or a higher mammal, what we have to do is we can do it by a method called somatic cell nuclear transfer. So this is the method which we have adapted for creating or producing Injaz, which is the world's first cloned camel. The same method which has been used for production of the dolly, which was the world's first cloned animal. Here, some thing about the somatic cells, because it is something related to somatic cell and nuclear transfer. So what does the somatic cell mean? Somatic cell is any cell in the body of an organism which is other than the reproductive cells. As we all might be knowing, reproductive cells are those cells like spermatozoa or the egg in the body of an organism. So if we see those reproductive cells, the spermatozoa or the egg, they have half of the chromosomes, half of the DNA of an animal. So the one half from the spermatozoa, the DNA, one half of spermatozoa, one half the egg, they unite together, combine together to give rise to a new offspring. So the offspring we get is having the genetic material half from the one parent and half from the other parent. Whereas if we see the somatic cell we use for the nuclear transfer, this is having the whole genetic material comes from only one organism, that is the donor animal or the donor cell. Nucleus is nuclear, something related to nucleus, which means that we can say the nucleus is like the brain of the cell. So it has all the genetic material which is being required for the formation of the new life, formation of the new organism. This information is in the form of DNA, which makes us unique, which makes us different from each other, which makes us different from other species. And now coming to the actual topic, these are the major steps in the cloning procedure which are being adapted and which we have also adapted for creation of the Injaz. If we see, so many steps are involved, a lot of the steps. But when I joined here in 2003 in the reproductive biology laboratory in Dubai, I was quite surprised. No basic knowledge about any of these steps was available to me. No basic knowledge was available. Even if some studies had been done in the science of the camel reproduction science, either they were not being published or they were just not ready to share the information with us. So what I had to start from scratch. We had to study all the way how to collect the oocytes from the ovaries, how to measure those oocytes in vitro, and what are the methods then we should use to activate these oocytes after we produce the somatic cell nuclear transfer embryos, and how should we collect and store the cells? How should we culture these cells for 7 days in the laboratory so we are able to have the embryos which we can transfer into the uterus of the surrogate mothers? So because we had some information available in another species like cattle, sheep, goats, but you should be knowing that the information which is available in other species, usually that doesn't work. Each species is different. So the available information doesn't work in chemicals. We have to start it again and start standardizing each of these techniques one by one. So if we go to the original protocols now here again, you can see that the first and the foremost two important things what we need. Yep. So the first and the foremost things which we need for the creation of the cloned embryos and the cloned animals is the oocyte or the egg from an animal, and the most important thing is the cell from the particular donor which we have to clone. So for these two, how we collect the donor cells from the animals which we have to clone? We can collect the cells from ovarian cells. We can have testicular cells. People have used even blood cells. They have used skin cells, and quite recently we have used the cells. The latest clone which we have produced this year has been produced from the skin cells of an animal. Skin cells, it has made the process quite easier for us because skin cells can be taken quite easily from the ear of an animal, and it's not that invasive method. After we take a sample of the skin, what we do is we cut this piece into small pieces, culture it in a culture plate till we get some cells. We have to culture them till we have sufficient number of cells available to us, and then we either freeze these cells for the future use or we have to use them for the nuclear transfer. But before using them for the nuclear transfer, we have to treat these cells because these all cells, what we have now, you can see these cells somewhere here, they are multiplying cells. They multiply quite frequently. And what we need for the somatic cell nuclear transfer or for producing the clone embryos is that these cells should be in a very dormant state. They should not be multiplying. So we feed these cells, rather we can say in our terminology, we starve these cells. We provide them with the nutrients that are only sufficient to keep them alive, and they don't multiply for about 3 to 7 days, and after that we see that these cells are quiescent and we can use them for the nuclear transfer. Then another raw material is the oocyte or the eggs. Where from to get the eggs? In case of the animals, we get eggs either from the live animals or we can get the eggs from dead animals, either whether they have been slaughtered or they have died naturally quite a few hours after their death. If you see in here, in case of these are the ovaries we get usually from the slaughterhouse material or we get from a dead animal. We can excise the ovaries out. So what you see here is structure which is like a fluid-filled structure. This is the follicle. This is the structure which contains the oocyte. What we have to do is we have to aspirate this follicle, the fluid which is in this follicle out by a syringe which is attached with a vacuum pump. And then in the fluid we have to search under stereo microscope to look for the oocyte. This method is usually being adapted for all the mammals or for the other animal species, but just to our surprise, to my surprise, this method didn't work that well in camels because what I saw is that the oocytes are attached to the wall of this follicle. When we aspirate the fluid, we get only the fluid, we don't get the oocyte. So we have to develop another method where we excise these follicles out in a tube and then under a stereo microscope, put them in a petri plate and tease them apart under a microscope and locate the oocytes and scratch them out from the follicle. How to collect these oocytes from the live animals? We have used the method called ultrasound guided transvaginal ovum pickup. If you say, again, in case of camels, it does not work as is being done in other species. We have to devise a method where the animal is made to sit down in the sternal recumbency and and ultrasound probe goes into the transvaginal. And I have tried to make a sketch to make you understand how actually we do it. So with one hand, taking hold of the ovary, you can see this is an ovary with so many follicles there on it. With the one hand, taking the hold of the ovary, another hand, the probe goes through transvaginal, and with the probe, there is a needle attached to it. What we see on the ultrasound screen machine is something like this. So these are the follicles which we have to aspirate. You can see somewhere the needle is here. So needle goes into each follicle, and we aspirate this fluid using a vacuum pump. Then again the same method, searching oocytes under the microscope. This is something what we get. So these are the immature oocytes. Why we call them immature? Because these are the oocytes at this stage. They are not ready for starting a new life. We have to mature them before using them for nuclear transfer or before using them for any of the in vitro embryo production technologies. How we do that? So we go now by step. You see, we have to mature these oocytes, and how we do that? Something after 22, 24 to 28 hours. Again, the problem here in camels, in other species, usually oocytes have been matured. Usually for 22 to 24 hours, it takes them in the lab to mature. And like this, where these cells around these oocytes or the eggs, they get loosened, and what we get is something like this, the oocytes which are now released, and we get something like this, where you can see a protrusion on the periphery of these oocytes. This we call as polar body. When we see this polar body in an oocyte, we say now this oocyte is mature and it's now ready to start a new life, and we can use this type of the oocyte for nuclear transfer or for any of the in vitro embryo production technologies. In camel, we have seen that what literature was available, it takes about 36 hours. When I use this for 36 hours, no embryos. I use 24 hours, no embryos. So we have studied again from scratch how much time it takes for these oocytes to mature, and what's the best time when we should use these embryos. So we came when analyzed our data, it was 28 to 30 hours, totally different from other species. If you see here, this is what happens to these oocytes at the nuclear, in the nucleus, in their nuclear level. Something chromatin is something like this when they are at this stage. When we say them, they are immature. So afterwards, within a few couple of hours, you see, they start arranging themselves, making into pairs, and each chromosome of a pair gets segregated to the opposite direction with the filaments, and within some 20 hours, you see both of them getting separated to each other, from each other, and after about 28 hours, what we see is this is a chromatin 1 and this is chromatin 2, which is outside here in this polar body. Something what you see like this. This image has been magnified over 300 times. So this is an oocyte now held with a pipette, which is one side. We call this pipette as a holding pipette. And this is with a vacuum, creating a little bit of vacuum. It holds the oocyte to it, and this is the injection pipette. We know that this is a polar body, so chromatin of this oocyte is somewhere here, but we need to visualize it. How to know where this chromatin is? Before we suck this ooplasm out of it, we give a very quick exposure to the ultraviolet light, maybe a few seconds, to locate we know the chromatin is somewhere very near to it. So what we do is we suck it out, but double check it under again the fluorescence microscope, again the fluorescence, to see whether really we have removed the chromatin or not. Then we take a cell and we inject the cell into the perivitamin space of this oocyte. I have a very small video taken last week. I hope it works, of this whole procedure. Yeah, you can see here now, this is the holding pipette, holding the oocyte. So with this injection pipette, we're trying to rotate this oocyte so that we have the polar body in a 12 o'clock position, because all these oocytes will not be having, this was a lucky shot, we are having the polar body in 12 o'clock position. You see, I have tried to see, localize where the chromatin is, so I know they are somewhere here now. So what the next step will be, to go there penetrate this membrane called the zone of pellicula, which is a very hard covering of the oocyte, and suck this ooplasm here, maybe about, should not be more than 10% of oval of this ooplasm. So we take this oocyte out from the field because we have to see, we have to double check whether really we have removed the chromatin material from this oocyte or not. So we just keep it there. We see under ultraviolet light again, and you can see both of these structures are here. So we are now sure that yes, this oocyte has been completely enucleated. There is no nucleus remaining inside now. So this is a waste material. We don't need it anymore. So what we have to do is we have to go somewhere the cells on the other side of the drop. We go to that side of the drop. This. Come on, it's somewhere here. So you can see these cells we have kept somewhere on the other side of the drop, and see these are the cells. See, this is also a single cell, and this oocyte is also a single cell. So the biggest cell in the body is this oocyte of an animal. What we try to see here is try to locate a very good cell. We should not be too big, we should not be too small. Somehow average size, having a good, good ooplasm, no breakage in the membranes. So what I have taken the cell into the pipette. We go to this oocyte again from the same hole where from we have tried to remove the chromatin material from this oocyte. We try to locate the same hole. If you can see, it's somewhere here.
I was thinking, Dr. Nisar, if in case of some of us audience is medically squeamish, it might be good to go through the procedures a little quicker, and then we can get the questions to you about what the cloning might impact.
Yep. So they are here. The cell is here in the periphery. So the next step is now the cell is there in the perivital space of the oocyte. So what we need to do is we need to fuse these cells with the oocyte. How to do that? For that, what we need is we need a stereo microscope attached with this is called a electrophorator, which provides the electric current at the specific time, specific voltage, and specific duration. So this is something, if you see in a closer view, something like this, where we have two electrodes having about 0.5 mm distance between them. So we keep the oocyte in between these two electrodes, something like this. You see, the oocyte something like this with the cell exactly on perpendicular to these electrodes, and we cause a current which should be something about 100 volts for 15 microseconds. So what happens after that? These two cells, they fuse each other. The nucleus of the cell goes into the oocyte. And the most important step then is we have to activate this oocyte now, because this cell is a somatic cell. It's not a reproductive cell. So we need to activate this cell, saying it, come on, you are no more a somatic cell, you are a reproductive cell, and you have to start a new life. If you see in normal in vitro fertilization procedure, what we do is, when a spermatozoa penetrates an oocyte, a sequence of chemical reactions occurs. These chemical reactions activate this oocyte, and because of that activation, the chromatin of the oocyte and the chromatin of the spermatozoa, they unite together and they form a zygote, which starts a new life. But here, in case of this nuclear transfer, what we have is a somatic cell, which is not activated in any case. So for that, what we do is we use a cocktail of chemicals for activating this oocyte, for activating the oocyte as well as the nucleus. After we activate this oocyte, the crucial step is how to culture these oocytes now, these reconstructed embryos, because we need to culture them for 7 days till they are at a stage when we can transfer these embryos into the uterus of a surrogate mother. For that, also, it was a very tough job for quite a few years. We had to try to develop some media, some methods, some techniques to do that. And this is what is like we call an incubator, which simulates the uterus or the womb of a mother where we incubate these reconstructed embryos. So these are the culture dishes inside this incubator where you see we keep our reconstructed embryos into these culture dishes for 7 days. What happens to this nucleus which we have fused with the oocyte now? If we see after a couple of hours, all the cells they start multiplying, and what we get is called a morula with many cells in it. Within day 4 to 5, we see that there is a fluid-filled cavity forming in it called, at this stage we call this a blastocyst, and after 6 to 7 days, this embryo develops so much that no longer this membrane no longer can hold it into it, and it ruptures it, comes out of it. We call that these embryos are now hatching out of it. So we have hatched blastocyst, something you can see here. So it is this stage of these embryos now which are ready to be transferred into the uterus of a surrogate mother. What we do is then we load these embryos into straws and something like this, transfer them into the uterus of an animal. So you can see here, the pipette goes into the uterus and these embryos are then transferred there. If we are lucky enough, after day 15 to 20, what we see is an embryonic vesicle formed, and then it was the time of injas. After 30 months of gestation, what we got is something like this. The animal was in labor, and injas was born on April 8, 2009. So, 3 more minutes, please.
Just want to give some time for questions for you as well, that's all.
Trying to get up on her, I'll go quickly through it. When she was going, this is our second clone camel now, which has been produced from the skin cells of a very elite bull called Sogan. That's why it's called pink Sogan. So why are we at present? These all steps I don't want to go through, but I want to say that our efficiency is as good as in other animal species. It's not that good. We can say it's still low. What we have done is so far, you can see we have transferred in these 2 experiments about 139 embryos. What we have got about into 79 recipients got 19 pregnancies and only 2 live births so far. What are our future plans is for preserving this, using this technology for preservation of the genetically valuable animals. Like we have good milk producers, we have racing champions, and we have males of very high genetic merit here. And using this technology for interspecies embryo transfer for preservation of the critically endangered species. Even in camels, we have one camel species which has 2 humps, which is called a backbone camel or double humped camel. Only few of those animals are now left in the wild, and we want to use this technology for preserving them and for preserving some of the wild animals here, particularly in the Gulf region, like so many animal species we are having here. For using this technology for embryonic and induced pluripotent stem cells, everybody nowadays is talking about stem cells, so they are being used for treatment and all sorts of for the disease and other conditions. Maybe in the future, we're going to do some experiments on producing transgenic camels by trying to modify the genome of these cells to see if we can produce some pharmaceutical proteins in the milk of such animals. Maybe in the future, we may have some milk from such animals which can be used for treating some diseases. And finally, I just wanted to acknowledge His Highness General Sheikh Mohammed bin Rashid Al Maktoum, our ruler of Dubai, for his moral and financial support, Dr. Ledida, our administrative director, and all the team at the Camel Reproduction Center, whether they are working in the laboratory or they are working out with the camels. Thank you very much for your